Hi, I'm Janine Cody, Director of the Chromosome 18 Clinical Research Center, and I wanted to share a little information about how to make a personalized syndrome description. And this is a little background about why you might want to do that. So the chromosome 18 conditions we group into 5 different groups, one being 18q-. Individuals with 18q- have a perfectly normal copy of chromosome 18 shown in this picture on the left, and their second copy of the chromosome has a missing piece from the long arm. You can see in the picture the chromosome has a constriction somewhere along its length, and it creates what looks like a long arm and a short arm, and the long arm is called the Q arm. So someone with a deletion of one copy of chromosome 18 of any size from any region on the long arm has 18q-. Likewise, someone with a deletion of any size from any region on the short arm, called the P arm, has 18p-. And in this group, half of the people are missing the entire short arm, and the other half with 18p- are missing just some portion of the P arm. So there's a lot of variability. Next, there are people with ring 18. So they have a normal copy of chromosome 18, but their second copy has formed a ring. And in order to form a ring, missing genetic material needs to be deleted from each end of the chromosome, allowing the ends to be sticky and forming a ring. And as you can imagine, there then would likely be missing genes from both the P arm, the short arm, and the Q arm, the long arm. And the amount or number of genes missing are different from one person to the next. Then there's people with tetrasomy 18b. And those individuals have 2 copies of chromosome 18, perfectly normal copies, but they have an extra chromosome. And that extra chromosome is made up of 2 short arms, 2 P arms, and hence they have tetrasomy 18p. And almost everyone with tetrasomy 18p has exactly the same chromosome content. But there are a few people who have a little extra material from the Q arm as well. Then there's trisomy 18, which is by far the most common of the chromosome 18 abnormalities. All the others are ultra-rare conditions. Trisomy 18 is much more common, it's much more well known, and many groups have focused on trisomy 18. And so our focus has really been on those individuals with partial trisomy 18. So they may have 2 perfectly normal copies of chromosome 18 and a third copy with a deletion of some part, the short arm or the long arm. Or they could have an extra piece of chromosome 18 from any part of the chromosome stuck onto one of the other chromosomes in the human genome. So again, the trisomy 18 group is not only people with 3 complete copies of chromosome 18, but people with partial trisomy 18 as well. And in order to help families understand the consequences of one of these chromosome 18 conditions, we've generated management guides, and there's also information for each syndrome or each condition on the chromosome 18 registry website. But from the explanation I just gave, you can see that any one of these syndrome descriptions is going to be very global, and much of it may not apply to any one specific individual with one of these conditions. For example, someone with an interstitial, meaning out of the middle, deletion of 18q is not missing the gene that causes one to have narrow ear canals, which is way at the bottom tip of 18q. So information about that wouldn't be relevant to someone with an interstitial deletion of 18q, just as an example. And from all of this, and this is a slide, is in almost every talk I ever give, anything you could do to this picture of all these human chromosomes can really happen, and it does really happen. If you could cut it up with scissors, glue it back together, or copy it and cut it up and glue it all together, it can really happen, and it does really happen. Many, the majority of combinations aren't compatible with life. This is the cause of the majority of miscarriages, especially early miscarriages. But some are compatible with life, and those are the chromosome 18 conditions. And as I said, we tend to group them into these 5 groups, knowing that each group is quite heterogeneous. So what do we do after we have global syndrome descriptions? How do we get more specific? Well, what we do is take a gene-by-gene approach to understand what the consequences of any one person's deletion or duplication might have for them. And there are 263 genes on chromosome 18. There are 67 genes on the P arm, so those would be genes affecting those with 18p minus or tetrasomy 18p or partial trisomy 18p. There are 2 regions of the long arm. There's one region right in the very middle of the long arm that nobody has a deletion of. Obviously, people with full trisomy 18 have a duplication of it, but nobody has a deletion of it. So there are 2 sections of the long arm, one with 87 genes, the proximal region, and one with 102 genes, the distal region of 18q. But the important thing to remember of all these 263 genes is that most of them, when you have 1 copy instead of 2, or 3 copies instead of 2, or even 4 copies instead of 2, they're just bystanders. They don't care. The body functions just fine with 1, 2, 3, or 4 copies. It doesn't matter. All systems are fine. Everything's working. But there are a few culprits. And they're the ones we've got to figure out which ones those are and learn to ignore what the function of the other genes are. So we call those bystanders dosage insensitive genes. So that's the term we'll be using going forward, dosage insensitive genes, to the genes that really don't make a difference when all there is an abnormal number of copies or an abnormal dosage of those genes. What we want to focus on are the dosage-sensitive genes, the culprits that are the cause of the problems. And a lot of effort has gone into trying to determine which are which. That's a major focus of our research and has been since we first began. And we have divided those culprits, the dosage-sensitive genes, into 2 groups. The ones we refer to as causal are ones that if you have a deletion or a duplication, 50% of the people with a deletion or duplication of that gene will be affected with whatever problem it causes. There are 16 such genes on chromosome 18. But then there are the low penetrance genes, those genes for which you have a deletion or duplication. Only less than 50% are affected with whatever problem that gene causes when it's in an abnormal dosage. And there are only 9 of those genes on chromosome 18. So when you think about the 263 genes that possibly cause a problem and realize that only 16 cause a problem in more than half of the people and 9 in a small number of the people, you realize that the majority are the bystanders. So how do you make an individual syndrome description for yourself or a loved one with a chromosome 18 condition? So we've created a website that you can use, and here's our website for the Chromosome 18 Clinical Research Center. You go to the part that says Chromosome 18 Gene Dosage Map. And when you go to that page, you'll see that there are 3 types of maps. And so let me explain the one you really want to care about. The first ones here are the gene dosage maps. There's one for deletions and one for duplications. Those maps are what I would call a little more geeky. They describe what we know so far of the role of every single gene on chromosome 18 with regard to what it does when you have an abnormal gene dosage, meaning you have 1 copy instead of 2, or 3 or 4 instead of 2. But, as I just told you, most of those genes really aren't going to be of consequence. They're dosage insensitive. So a map that shows every single gene has a lot of stuff you don't need to worry about. Then there are the phenotype maps. And as I said, we spent a lot of time trying to figure out which genes are the causative genes, the dosage-sensitive genes, and which ones are not. And in that process, we've narrowed down regions of the chromosome associated with different conditions or different characteristics of these conditions. We haven't figured out just which gene, but we've narrowed it down to, in some cases, very small regions of 3 or 4 genes, and that's a focus of our ongoing research. But even though we don't know the gene, it is helpful to know which region is associated with which characteristic. And so the phenotype maps also show every single region associated with a characteristic, an abnormal characteristic. Some of those might be related to gene dosage and some not, and some we don't know. But here's what you really want to look at. These are the clinical dosage maps. These maps show only the information for the genes that we think have a clinical consequence. So right away you've eliminated all that other stuff you really don't need to worry about. And it also includes, in the very same map, the phenotypes as well. So this is what you want to look at. And there are 2 of them here. It's called the deletion map, and one is the duplication map. And I'm going to show you what each of those look like when you click on those. So when you click on the deletion map, again, just a reminder that it only includes those genes that are dosage sensitive and ignores the rest. So when you click on that, you get a map that looks like this, which looks kind of complicated at first, but here in the middle we have the diagram of the chromosome, which you've seen many times. On the left-hand side are all the phenotypes or characteristics and their associated gene that we've identified as being causal. And you'll notice that some of those are in bold font and some are in normal font. The ones in bold font are the ones that we call causative and cause an issue in more than 50%. Now, I need to tell you that most of those more than 50%, they're still only 60% or 75%. There's only one gene called TCF4 that results in the characteristics of Pitt-Hopkins syndrome that affects 100% of the people with a deletion of that gene. None of the other genes affect 50% of the people. And those genes and phenotypes listed on the left that are in normal font are the ones associated or not causative. They're low penetrance, affecting less than 50% of the people. So many of those are 10% of people affected or 25%. So some are pretty low percentages. On the right-hand side, each box indicates the region associated with any one of the phenotypes shown here. And again, you see some of these boxes are dark gray and some are light gray. And again, the dark gray boxes are those phenotypes that are found in more than 50% of the people with a deletion of that region. The light shaded boxes are the ones that indicate a region that causes that condition in less than 50% of the people. What's interesting here, though, is that on the phenotype side, where we don't know exactly which gene, most of those are the light-colored gray. They're the low penetrance, which is part of the reason it's been harder to identify a causative gene. And if you click on this, actually what will happen is you'll go to another page that looks just about like this. That other page is actually not on the UT Health website. It's on the Chromosome 18 Registry website. So then when you click on that in any one location, it will take you to a page that looks like this. Here I've clicked on the one specific gene on 18q, and it took me to the page with information about that gene, the position of that gene, and all the references for what we know about that gene and its effect when you have an abnormal number of copies. So this information is also on the Chromosome 18 Registry website, and the reason is my staff can go in and edit this anytime they need to, as opposed to on the Health Science Center website where we have to put in a work order for some minor change that 2 weeks later will probably get done, maybe, hopefully correctly. So it gives us more control and more direct and quick control over what happens, which is why you'll notice that it's on a different website. Likewise, the exact same thing is there for a gene dosage map for the duplications. What you can see, though, is we know a lot less about the duplications and the specific genes involved in trisomy 18. This is a major area of work for us now. There are many fewer people with small duplications of chromosome 18, making it harder to identify small regions. Also, mouse models and other techniques that we use to understand deletions are much more problematic to give us the kinds of information we need on duplications. So in general, it's a much harder nut to crack, but we're working on it. So there are lots of resources for people with chromosome 18 conditions. The registry website has revised condition descriptions that are really in lay language, and many introductory videos, hopefully not too boring, but hopefully informative about these conditions. And the Chromosome 18 Clinical Research Center website has, as I've already said, the physician management guides for each condition, and of course the gene dosage maps, which I've just showed you. And there are lots of monthly videos and articles explaining some aspect of the chromosome 18 conditions, answering many of the frequently asked questions. Should you have other questions, which are often the basis for me to figure out a new video I need to do, if you have the questions, somebody else will too. So it's a great, not only will help you get an answer if you ask a question, but will help other people get answers too. So if you have questions, just email us at chromosome18@uthscsa.edu, or you can go to our website and find all the information there. And just as a bit of a teaser, if that's possible, you noticed in the slides I showed about the dosage-sensitive genes that there's all this information in dark gray about dosage-insensitive genes, which can explain some of the variability from one person to the next, the rare aspects of the chromosome 18 conditions. And that is in another video about why there's so much variability from one person to the next, even though they have the same condition. So, thank you so much for your attention. I look forward to meeting every single one of you in person one day. Thank you.